Stephanie. I'm an aquatic research biologist with the Ontario Ministry of Natural Resources and Forestry. And today I'm going to be talking about using community eDNA metabarcoding as a detection tool for aquatic invasive plants. So this started as my master's research back in 2016 at Trent University, and now working with the MNRF, we're doing some follow-up work. So this is just an overview of our team. So Chris Wilson, as most of you know, is our research scientist. There's me, Christine, our senior tech, and then Kaylee and Jess are our resource technicians. So a lot of our projects in the lab focus on fish genetics and genomics. We are also involved in a lot of eDNA projects that focus on different types of aquatic organisms. So we do some single species qPCR testing, looking for presence or absence of things like invasive carp or invasive aquatic plants like water soldier and hydrilla. And we've also been involved in and led some projects for community surveillance and metabarcoding for fish, unionid vessels, and aquatic plants. And that's what I'm going to focus on for the rest of the talk. So as I mentioned, this started as my master's research at Trent University with Drs. Joanna Freeland and Dr. Aaron Schaefer. So previously to this, the only methods for locating and identifying aquatic plants, including invasive species and species at risk, was either visual surveys or using eDNA barcoding, so single species detections. There are some limitations with both of these methods. You might have some hard to access areas, things like clarity of the water, water depth. The plants can be submerged or floating. And there's a lot of issues with misidentification, specifically with plants. There's a lot of cryptic species, a lot of hybridization that presents some challenges unless you've got an expert taxonomist to identify your plants. And then barcoding can be a great tool for single species, but it only allows you to target one or a small number of species at a time, and essentially leaves you blind to everything else that might be present. So our goal is to investigate the feasibility of using metabarcoding to characterize aquatic plant communities from eDNA water samples. So the first step was designing primers. I won't go into detail. You've heard lots about designing metabarcoding assays today. We targeted 3 regions in the plant genome, so MATK, RBCL, and ITS2. We looked at DNA sequences from publicly available reference databases for common species from our kind of local areas, the Kawartha Lakes, Trent River, and the Rideau River systems. That included a lot of invasive species. And then looked at alignments of those sequences to look for appropriate metabarcoding regions. And then we tested these primers on single species to start with. And then we tested them on 2 types of eDNA samples, some mock communities and some wild samples collected from 2 regions in southern Ontario. So the samples were created or collected, the mock community was created from plant clippings from a number of species, and then filtered and extracted. So these are the mock community results. The table that I have on the right, you'll see there's 5 columns. So there were the 3 assays that we designed targeting MATK, RBCL, and ITS. And then we also tested 2 metabarcoding assays, that were designed for terrestrial plant metabarcoding from soil samples. So we wanted to see whether those would also work for aquatic plants, or if there's some more sensitivity for assays that were designed specifically for aquatic plants. And then here, so a black square represents a species level detection, and a gray square represents a genus level detection. So you can I just highlighted a couple of the best performing assays in terms of number of species level or genus level detections, and then just to show that combining the results from the top 3 performing assays does slightly increase our number of species level detections. And then this is just the same results. I've just highlighted the invasive species or species of concern in Ontario, just to show that we are able to identify a number of them with multiple assays. This is the same results, just looking at the relatedness of the species that were included. And again, black is a species level detection and gray is a genus level detection, just to show that our detections were not biased in one particular taxonomic group. It's spread across the taxonomy of species included in our mock community. The next step was to test the assays on some samples that were collected from the Black River that feeds into Lake Simcoe here in Ontario, and an additional 5 sites from Seymour Lake, which is part of the Trent River and the Trent Severn Waterway, So we identified 17 and 14 species at both of those locations, and then I just again listed some of the noteworthy detections of invasive species or species of concern in those areas. So now working with the MNRF, we've been doing some follow-up work. We're testing ORBCL2 on more sites and more replicates. We've got 44 sites right now. This work is still in progress. We decided to start with ORBCL2 for some of the follow-up work because it identified all but one species from the mock community, at least to the genus level, and also identified the largest number of species from the pilot sites. The sites that we're looking at now, these 44 sites, were actually collected specifically to monitor for water soldier, but we're also applying metabarcoding to see what else we can find. And it's an ongoing project to see the effects of plant removal and herbicide treatments. And then again, I just have some of the noteworthy invasive species detections, including water soldier there from metabarcoding. So there are some limitations, challenges with plant DNA in general. Plants have some pretty complicated genetics, so that makes assay design a little bit tricky. Genetic variation between different species is not always greater than members of the same species, so that makes both the primer site selection and the amplicon design, I guess, a little bit tricky. And then there's also a high level of hybridization of plants. And then, as you saw maybe in the mock community results, often we are getting the genus level identification, not always species level. So those are some challenges. However, it might just mean that using a multi-assay approach, like I showed, if you combine the top 3 performing assays, and you do identify more species level, if that's the goal. It doesn't offer insight into abundance, but again, you could follow up if you have more questions about a particular species that you find in your metabarcoding data. Follow up with something like qPCR that's a bit more sensitive. And then it also relies on having, and we talked about this a lot today too, having complete and error-free reference databases or libraries when you're comparing your sequences for species identification. So if there's errors or gaps in the reference database that you're using, then you might end up with gaps or errors in your data as well. So, however, it can be a great tool to monitor biodiversity over time. This can include looking at co-occurring organisms, looking before and after alien species invasions. You can use it beyond plants. As you all know, with eDNA samples, you have DNA from everything that's theoretically everything that's in that environment. You can use the same samples for multiple different queries. And then going forward, it'll be really important, again, as I was just talking about, to establish a more comprehensive baseline of these reference libraries for metabarcoding queries. So getting voucher specimens, making sure that you've got expertly ID'd plants. It can be used for early signs of invasion. Again, with metabarcoding, you don't have to have previous knowledge of what you're looking to find. You can combine multiple assays to increase those number of species detections that you're getting. And again, as I said, you can use targeted inquiries like qPCR detections to get some more information. Okay, I think I'm a little under time, but that's all I have for today. So if anyone has any questions.